Hi everyone. In this video, we're going to have a look at the nucleus and ribosomes and their job in producing proteins from the cell's genetic instructions. The nucleus is often the most prominent organelle in an animal cell, as shown on this model. Houses most of the cell's DNA and acts as a control center for the cell by providing instructions to make all of the cell's proteins. The nucleus is separated from the rest of the cell by a double membrane called the nuclear membrane or nuclear envelope, and we'll look at this a little more in the video covering the endomembrane system. Most of the time, a dense spot called the nucleolus can be observed inside the nucleus, which is where ribosomal RNA is produced. The ribosomal RNA will then be transported to the cytoplasm along with certain proteins, where the final stages of ribosome assembly will take place. Chromosomes can be either circular, as commonly found in prokaryotes, or linear, like these eukaryotic ones. In eukaryotes, DNA is packaged into chromosomes by wrapping the DNA around proteins and folding it through a complex multi-level process to enable all of the DNA to fit into the nucleus. The sequence of the bases in the DNA function like the letters of a word, with the number and order of bases specifying the order of the amino acids to use in protein synthesis. Ribosomes are very small structures made of complexes of ribosomal RNA and protein, and they can be found in two locations within a eukaryotic cell, bound on the surface of the rough ER or free in the cytosol. Ribosomes can and frequently do switch from one location to another, depending on the type of protein to be made, which we'll look at more in a later section on gene expression. Let's zoom in to this free ribosome to have a look at its function. The ribosome is made up of two pieces, the small and large subunits, which work together to translate the sequences of bases in messenger RNA, shown here in purple, into a sequence of amino acids. The amino acids will be brought into the ribosome by molecules of transfer, or tRNA, shown here in teal, and the ribosome will help to catalyze the formation of peptide bonds between one amino acid and the next, creating the primary structure of the protein. Thanks for watching, and please make sure to check out the other videos in this section.